Again, let me thank the organizers for having invited me back to India. It's a wonderful opportunity. So I'm going to change directions a little bit because actually I do think that metronomic therapy is way more than just the vasculature. And in fact, I'm going to focus on the immune system that we've kind of touched on multiple times today. I don't have any specific conflicts for this talk as the drugs haven't entered clinical trials yet, but I'll point that out momentarily. So remember that the whole concepts of metronomic were originally designed to target angiogenesis. In fact, as Bob pointed out yesterday, the original title for this field was anti-angiogenic therapy, not metronomic therapy. And again, I think there's now a clear recognition that the microenvironment, including neovascularization, is much more complicated than we had originally thought when it was first identified. So what I'm going to do is talk about this concept of when we target the tumor cell, obviously the damage to the tumor cell influences the microenvironment, and while our job as oncologists are to kill as many tumor cells, as I'm going to show you, killing tumor cells actually stimulates other tumor cells in what we now refer to often as the double-edged sword. And there is a process in the body that naturally is designed to control this process and called resolvins. And obviously, both the tumor regulates some of these, and as I'm going to show you, we may be able to pharmacologically modify these to better regulate the microenvironment. So obviously, I came from Judah's program, and Tim Browder, in fact, was a pediatric oncology fellow that was there before me that actually approached this from a very oncologic point of view. So the premise of virtually all oncology training is of a little bit is good, more is better, and in fact, it really gave rise to the whole concept of high-dose chemotherapy and stem cell rescue, is you basically go to beyond the toxicity limits of the human being. And that's not surprising that that permeates everything we do. And at the time that there was this discussion about exactly how chemotherapy works, if chemotherapy kills dividing cells and the angiogenic models were correct, then one would anticipate that you should be killing the endothelial proliferating cells that develop the angiogenesis as much as the tumor cell. And so the question that got asked is, why isn't chemotherapy effectively anti-angiogenic? And I'm going to talk about that momentarily, but I wanted to just kind of summarize that, again, we now see this in a much larger compartment. Obviously, when metronomic therapy first came around, it was really all targeted on the concept that if you could inhibit endothelial cells on the assumption that that's what angiogenesis was all about, you would be successful. Obviously, there are now a large number of small molecule or antibodies that inhibit neovascularization. There are all kinds of new targets, basic FGF. You heard about the angiopotents, for example. But one of the things that struck us that I'll talk about in a moment is the recognition that all of the things that target the endothelial cell also, it turns out, target the microenvironment, in particular the inflammatory and immune response. And in fact, many of the original metronomic-based drugs, things like celecoxib, thalidomide, are actually immune modulatory agents as much as they are anti-endothelial. So again, in the original concept of, as oncologists for the first 30 or 40 years, when everything we did was based on killing tumor cells, which is why we maxed out on chemotherapy, it's why we maxed out on radiation therapy, that made a lot of sense. That was the problem. And then obviously when Folkman came up with this concept in 1970 of, actually it's not just about the tumor, that in order for a tumor to grow, it needs this neovascularization, is why I think we've been so focused on the anti-angiogenic component of this. And unfortunately, I think this is one place where Judah perhaps underestimated the complexity of the system. And I think now we recognize that, although, sorry, there's something not showing up here, in addition, on the far side, there should also be now the endothelial cells, the stroma, the migrating bone marrow-derived cells, the inflammatory cells. So we now recognize that the tumor is not made up of two compartments. It's actually made up of the tumor cells that have a certain microenvironment and all of the cells of that microenvironment that basically regulate the process. Well, to kind of take a slight detour for a couple of slides. So if we go back to the whole concepts of inflammation, when you injure yourself, cut yourself, scrape yourself, obviously you get a little bit of your skin bacteria into the wound, and that starts an inflammatory response. And the human body over the last billion years, or at least million years, has developed a relatively effective way of managing that. It induces an inflammatory response with the recruitment of mononuclear cells that come in, basically clean up all of the bacterial damaged cells, and then through a series of monocyte and macrophage evolution, you basically clean that mess up. And in doing so, you go from an acute inflammatory response to one in which everything goes back to normal. And obviously, that's what we hope happens with every infection. Obviously, there are lots of circumstances where there's incomplete modulation of this process through a variety of the elements in the pathway, including the polymononuclear cells and or the monocytes and macrophages, where you now basically get sustained inflammation. And for anyone that's been through medical school, there are entire chapters on inflammatory diseases that are a result of the fact that you can't seem to turn the immune system off normally. And this kind of chronic inflammation kind of highlights a number of different diseases that we won't go into today. Well, it turns out if you asked how does the body normally regulate this process, it regulates it through a group of molecules called resolvins, given the name because they obviously resolve the immune system, and they basically have two functions. The first is that they basically regulate the phenotype of macrophages, so that macrophages are sent in of a specific subtype that go in and clean up all of the mess in the area that's inducing inflammation. And they also have very potent anti-inflammatory responses that shut down the signal. So while they're getting the macrophages to clean stuff up, they're stopping the signal from being perpetuated, and it's those two things that eventually lead to that decline in macrophages and arrest of the inflammatory response. And so obviously the question is, where is that relevant in these models? Well, it turns out that there are now a number of resolvins that have been discovered. Typically the D series, D1 and D2 are the most well characterized, and the E series, E1. There are also these things called protectins and maricins, and they're basically all the same family with some very slight variations. Remember that when the angiogenesis field started, for the most people assumed that there was VEGF, that VEGF induced neovascularization, that if you could inhibit VEGF, you would basically cure all cancer. We talk a lot about bandwagons that we jump on and off, and remember that, you know, only a decade or 15 years ago, everybody assumed that angiogenesis would cure everything, and the picture that Bob showed today of everybody in the theater in the immunotherapy section, well, you could, if you had just backdated that, those were exactly the same things 10 years ago when everyone was attending the anti-angiogenic sessions and not something else. Remember, we did the same thing with siRNA. We did it with monoclonal antibodies. There have been a ton of these bandwagons that have gone forward. Certainly immunotherapy is one of the newest ones, but I suspect in and of itself will not be the total answer. At the same time, any system that has this many molecules regulating it, just like it turns out angiogenesis has so many different molecules that regulate the process, which is why it hasn't been easy to just inhibit VEGF and cure everything, suggests that these are going to be important in the control of the circumstance. So what are resolvins? They're basically just endogenous lipid autocoids that are basically derived from omega-3 fatty acids, which is why your mother was always telling you to eat all of that stuff that you didn't really want to eat. And it's because those turn out to be really good for you. And they basically, again, have two properties. They both suppress inflammation and they resolve via macrophages the cleaning up process that stops the inflammatory response. So now what I want to do is transition to this one other issue that was a really exciting brand new idea until, of course, we discovered that somebody had already thought about it in 1956 before most of the people in this room were even born. And unfortunately, it is somewhat true that most of the good ideas have already been taken. So it's actually referred to as the Revez effect, and I don't know if any of you have heard of it because it's not something you would have heard about unless you were fishing around publications back back in 1956. And what this individual did was he did a very simple experiment. He took a certain number of tumor cells and he injected them into mice, and obviously he eventually got some tumors to grow, and then over a period of time some of the animals died. And virtually everyone in the room that has a lab has probably done that experiment multiple times before. What he then did is he was a radiation biologist, and so what he did is he took some tumor cells and he irradiated them and killed them, and then took the same number of live tumor cells, mixed the two together, and then just injected them into the same sets of animals. And what he discovered is that all of the animals rapidly developed tumors and all of the animals died. Now, the difference between those two experiments is it's exactly the same number of live tumor cells. He was simply altering a little bit of the microenvironment in which they were implanted, and suddenly he could dramatically change the ability of the tumors to grow and kill the animals, and hence the naming of this phenomenon, the Revesz effect. Well, this is this concept of the double-edged sword. What this means is that if, as oncologists, our job is to kill as many tumor cells as you wanted, and it gets to Yaroslav's question of would you leave a piece of tumor behind, the answer is no, because if you've got some tumor cells there and you kill them with chemotherapy, radiation, whatever, the spilling of their guts basically provides the exact environment that will allow any surviving cell to grow much better, much more effectively, and kill you faster. And it's this concept of, as oncologists, we're trying to kill as many cells as we can, while in the very process of killing some cells, we're basically helping the tumor grow. So obviously it raises an important question from a kind of biologic point of view. Could you control this process and clean up the stuff that you've killed so that it doesn't have that stimulatory effect, using the system in the body that's already there to do exactly that? So again, this is an experiment that I suspect many have done. If you take, this is, I believe, Lewis lung carcinoma. So if you take 1 × 10⁶ cells, you inject them into the sub-Q of a mouse, you get tumors. They kill the mice rapidly. If you inject fewer cells, you still get tumors, but they take longer, presumably because the microenvironment's not quite so rich. And you can get to a dose that's basically considered dormancy. This is where if you inject 1 × 10⁵ cells, the cells are still there and they're still rapidly dividing. The issue is that because they haven't developed the entire cascade required for tumor growth, basically the cells just sit there. And in these animals, 6 months after they've been implanted, if you go in and take out the little area that you injected, you can still find the rapidly dividing Lewis lung cells. We'd always assumed that what's happening is that as each time a cell divides, one of the cells gets pushed beyond the neovascular capacity that the tumor has because it hasn't generated its own angiogenesis, so it basically suffocates, and you're in this equilibrium that prevents the tumor from getting bigger. And obviously this represents carcinoma in situ. So the whole reason for a pap smear is because you're basically discovering someone that's got cancer that if you simply take it out before it's figured out the second part of the oncogenic pathway would now be curable. So we asked the same question that the Revesz effect did, only this time instead of using radiation therapy, could you do the same thing with these dormant tumors in using chemotherapy-based approaches? So this is an example of a tumor, let's see, this is Lewis lung carcinoma with cisplatinum. So if you just take a whole bunch of dead tumor cells and you inject them into an animal, not surprisingly, nothing happens. There are no live tumor cells there at all. If you take just the small amount of tumor cells, you mostly get these small dormant little lesions that remain largely quiescent. If, by contrast, you take the same number of live tumor cells that didn't really produce an active tumor, but you now add in that tumor debris, you now start to get the tumor to grow. And if you add 3 times as much debris, it grows faster. And if you add 3 times as much debris, it continues to go faster. And you can now see by 15 or 16 days, all of the animals are dead of disease. And again, this is where all of the animals have gotten the same number of live tumor cells. It's the microenvironment that's being modulated. You can do this with a variety of other cells. We've done it in now 10 or 15 different cell lines. In this case, again, the tumor cells alone don't produce tumors. The apoptotic dead cells obviously don't produce anything. If you combine the two together, they nicely induce rapid tumor growths. If you treat them with cisplatin in this case, you don't actually see much of a response. That was a little bit of a surprise to us until we understood it's a bit of a dose effect. We'd put in so much debris, the cells were so stimulated that basically the cisplatin wasn't enough to overcome it. But this time, if you add in the resolvents, the things that clear debris, you can now potently inhibit the ability of these tumors to grow. Same thing, this time again Lewis lung, this time again with cisplatin. This is a slightly different animal model, as I think you heard earlier. These are very dependent on a variety of things, including strains. Here again though, apoptotic cells, no growth. Again, controls grow. Cisplatin in this case did have a little more activity, but again the resolvents can potently inhibit this process. You can do this in a variety of other ways. You can say, is this just a chemotherapy effect? And we've now done this with virtually all of the major chemotherapeutic agents, the alkylators, the mitotic inhibitors, and so forth. This is an example with a biologically based drug, Tarceva, where again, you see the same thing. I didn't put all the arrows on it. This is a case where you're now using tamoxifen in a breast cancer model. Again, using a hormone, you get the same thing. This is not dependent on the specific inhibitor you use. In this case, the inhibition is with the Morrisons instead of the resolvents, but basically the same thing. You can do all kinds of analyses to look at the downregulation of the inflammatory signals, in this case IL-6, IL-8, TNF-alpha, in which the purple staining is increased, and you can see as you treat them with the resolvents, you dramatically reduce the amount of that, and I'll show you some quantification of that a little bit later. You can also ask the question, does it absolutely require that you have added in the debris, since most humans that got their tumor didn't have someone standing over them with a big debris syringe injecting at the same time their tumor was developing. This is an example of animals injected with U87, U87 glioblastoma multiforme. Remember that one of the diagnostic criteria of glioblastoma multiforme is that the cells basically become apoptotic on their own. Palisading necrosis is one of the diagnostic criteria, along with vascular proliferation. And you can see that if you just inject glioblastoma cells, the assumption is that they're turning over so rapidly and producing their own debris that, again, you can potently inhibit them with resolvents without having even added any of the quote microenvironmental component that, as I showed you in the previous experiments. It actually turns out it's not just the inflammatory response, that actually these are two papers from Nature Medicine, one looking at the ability of the microenvironment to alter some of the developmental signaling pathways in the upper panel with Wnt, and in the lower panel alterations in BRAF signaling that also is one of the mechanisms by which you can become resistant to the BRAF inhibitor. So obviously the microenvironment plays a very important role, not just in the inflammatory response, but in a variety of other pathways that regulate the microenvironment in a way that's optimal for the tumor. So we've shown using, and I didn't show you most of the data, chemotherapy-induced debris promotes the tumor growth, and that resolvents can basically inhibit this process by what is thought to be its function on macrophages. And obviously, because it raises the question of whether these drugs can be used clinically. It came up with this idea, much like we used to talk about the angiogenic switch, could we talk about the inflammatory switch, in which when the tumor microenvironment is heavy with debris and the cytokines, you basically have a tumor growth environment, and if you can change that balance by reducing the inflammation and improving the amount of resolvents present, you could presumably result in tumor shrinkage. So this was the original hypothesis, that resolvents would act on macrophages. We had a little bit of preliminary data to suggest that that was true. It looks like what it does is it converts an M1 macrophage into an M2 phagocytic macrophage that then starts to clear the debris, and that's the mechanism for tumor growth. You could ask the question, if you take the debris from one tumor type, does it also have the same effect on others? And the answer is yes, but in a somewhat complicated mechanism. So in this case, this is Lewis lung carcinoma, and again, you can see that if you develop debris, in this case from Lewis lung carcinoma, you can see the different stimulatory effects on fibrosarcoma, on melanoma, on Lewis lung, on pancreatic cancer, and so forth. So there are differences, but they do all stimulate. The other thing you could do is you could say, well, cell debris is more than just cell debris. Does it matter whether the cells are apoptotic versus necrotic? And in fact, it appears that it does. Apoptotic debris appears to be a much more potent stimulator of the microenvironment than necrotic debris is by this and multiple other experiments of which I'm not going to show you due to time. But here's an example, again, of at the bottom with just debris alone doesn't do anything. With tumor cells alone, you don't get much growth. In the combination, you do. If you then said, okay, what about standard chemotherapy? Again, 5-FU and gemcitabine, you can see certainly inhibit some of the growth, but they don't stop it completely. Again, presumably by virtue of this double sword effect. If you ask what about anti-inflammatory agents like Celebrex and dexamethasone, again, they certainly seem to do better than even the chemotherapy does. But if you now use resolvins instead, in fact, they are by and far the most potent way of inhibiting that kind of tumor growth. Obviously, I've talked a lot about macrophages. These are just some of the experiments we did. So we took liposomes that were empty, and again, here the control with debris plus tumor cells, they grow rapidly. If you treat with resolvin, you can inhibit a lot of that growth. If now you add in the, instead of empty liposomes, those with clodronate, which basically depletes the macrophage component, you can now see that the resolvins no longer work. It appears that they depend on the presence of macrophages for their function. If you treat cells coming from just normal donors with these different resolvins, you basically change them from the M1 phenotype to the M2 phenotype, and this has been well documented in other systems, that again would suggest that that's the critical component in their activity. As I said, if you look at the ability of them to upregulate with tumor debris to upregulate a variety of pro-inflammatory and pro-angiogenic cytokines with cisplatin-induced material, again, you see exactly the kinds of pro-tumor stimulatory patterns that one would expect that you can suppress, again, with the resolvins. Obviously, we wanted to be sure that this was actually phagocytic in nature, so we took the B16F melanomas that actually had some of the melanin that you can then pick up on EM, and again, you can see on the arrows that the phagocytes are actually picking up those cells. That it does appear to be what's happening to the tumor cells. They're now, the debris is now being ingested. So finally, there are certain other mouse models that one can use. The mannose-binding lectin deficient mice is one where basically the phagocytes don't work properly, and because they don't work properly, they can't clear debris, and not surprisingly, in the transgene of those mannose-binding lectin mice, the tumors grow even faster, again, because once you get any inflammation within the tumor bed, they basically can't control it at all. We actually, in conjunction with one of the groups, in fact, the individual, the Sheehan group that discovered these molecules, there have now been a couple of the receptors, but not all of them yet, that have been discovered, including the ALX receptor for D1 and the R23 for the E1 resolvins. And that's important. So one of the things we did is we stained both macrophages and endothelial cells for the expression of both the D1 receptor as well as the macrophage markers, F480, and you can see the yellow says that they're overlapping. Hopefully there's no colorblind issue there since they're all yellow. And similarly for the endothelial cells, the markers are exactly where you would predict they should be. If you then take out a knockout mouse in which we basically remove the receptor for D1, you can now see that those mice, because they can't use receptor, they can't use the resolvins at all, basically they now have dramatic increase in the activity of tumor growth, again consistent with the models. if you just ask in the models that you guys had talked about earlier, where you've removed the primary and then ask what starts to happen to the metastases, again, you see dramatic upregulation in the number of METs in lung, liver, lymph nodes, and so forth in these knockout mice. So this concept of the inflammatory switch is one in which we believe that the tumor microenvironment, particularly in the context of radiation and chemotherapy, is idealized for the tumor cell, the surviving tumor cells, and that it basically allows them stimulation and eventual tumor growth and metastasis, and that obviously it's the ability to control that inflammation that would be an important part of therapy. Which doesn't mean that you would therefore stop using chemotherapy and or radiation. What it means is that if you gave those in conjunction with a resolvin, you could kill the cells but clean them quickly so that they no longer have the stimulatory effect that we believe they do now. And the good news is that the resolvins have already entered clinical trials for 3 major inflammatory diseases, which obviously is a much bigger market, or has been a much bigger market for the pharmaceutical companies. So these 3 trials have just started, which means we actually now have the opportunity to try and get some of these drugs into human clinical trials as well, which is something we're negotiating with the company. So in summary, there's a group of these molecules, resolvins, that probably most of you have never heard of before, that turn out to be major regulators of the inflammatory response. And the belief is that the inflammatory response is as important to the microenvironment as neovascularization as it is to all of the other components. And that as we think about the context of metronomic therapy, until we consider the entire microenvironment together, I suspect going at any one target is not going to be completely successful. And then I just wanted to stop by recognizing Deepak Panigrahi. For those of you that don't know him, he was actually the medical student that Judah picked up very early on in his career and convinced him not to complete his surgical residency, that there was much more important stuff to be done in the lab than in the clinic. And so Deepak basically never went back to his residency and became my postdoc when Judah passed away, and we've been doing all of these studies together. And then a couple of years ago, Deepak and I changed the circumstance where we now share a laboratory. So he is the co-PI of the laboratory and again has done a phenomenal amount of the work that I just showed you. Charles Sirhan is the person that discovered the resolvins and has made many of the molecules to them. And then these are the people actually in the lab, the postdoc, Yale, and then I'd say that Deepak does all the hiring, so the fact that there are 7 women in the lab to him. But again, it's been a remarkable group, and in fact, all 6 of them have either already now gotten accepted to medical school or are in their interviews for this coming year. And then just to recognize the group, including my group before when it was still part of the Folkman empire, and then the people that have been involved in the more recent work, as well as some of the funding, and I'll stop there.